All right. Welcome, everybody, to Proteoform Thursdays. I'm Jeff Agar. I'll be your illustrious host today. I'm going to pass this to Paul Danis for a minute and let them tell you a little bit about the consortium, and then we'll come back and introduce our speaker.
Great. Thanks, Jeff. Let me just give you a couple little updates on the consortium, some of the activities that are coming up, as well as other information you find useful. Um, so first off, we want to thank all of our sponsors who have been supporting us for a number of years: Thermo Fisher, Bruker, SciEx, and Lilly, as well as our supporters Agilent, Pfizer, Mass Spec Consulting, New Objectives, and Impro. And from these folks, we're able to do these kinds of programs and grow the consortium, grow the community. So if you see these folks somewhere, just tell them thank you. We appreciate the support. So one thing, just to give you a little update, some of you were able to come to the symposium in Hamburg last month. We had an exciting several days of activities and joyful fun and great science. And if you're interested, the book of abstracts is available on the website. If you want to go there, you can take a look and see some of the work that was presented. And I'm sure you could follow up with some of the authors if you really had interest in some of their work. And as far as upcoming events, next month we're going to have our vendors technology showcase where some of our key sponsors are going to be presenting some of the latest technology they have. I mean, it's tough to keep track of what the latest technologies are from all these. So we're going to get them together and say, all right, what's the offerings for top down and how can people benefit from these? So we'll have that next month, which will be kind of fun. And in November, we have Christian Bleiholder from Florida State University. And then in December, we have Jared Shaw from the University of Nebraska. So some great things coming up this fall. I hope to see you there. And now I'll just hand it back to Jeff. And hope you have an exciting, fun hour here. Thank you.
Thanks so much, Paul. Well, it's really an honor to introduce Jenny Brodbelt, a great scientist, a hero. Jenny has an amazing background. She's the Rollin Petit Centennial Professor of Chemistry at UT in Austin, and is also chairperson. That's a big deal, trust me, everyone. She earned her bachelor's at University of Virginia and her doctorate at Purdue with Graham Cooks. And then, if that wasn't enough, a postdoctoral fellowship with Mike Bowers. She's started at UT and she's been there the whole time. She's also the director of graduate education. And as I said, chair, she's the editor-in-chief of the JASMIS and the president of ASMS in the past. I think most of us in this room don't need to be told that she's the world's expert in UVPD, but in so many types of molecules, which I'm hoping we'll hear about. And Jenny has, I looked, 470 plus papers, pretty amazing. And I can't go through all the honors because I want to give Jenny time to talk. But in the last year, anyway, the ASMS John Fenn Award for Distinguished Contributions and the Thomas Medal. And with that, Jenny, I'm going to pass it to you. And thank you so much for being here.
Okay, great. Okay, hopefully everyone can hear me and soon be able to see my slides. Does that look good, Jeff?
It looks to me like you might need to switch screens.
Okay.
We're seeing your notes, and
Okay, that's always fun.
I know I can never figure that out.
I know. Let me try again. Any
That
better?
does it. That's it.
Okay, awesome. And here we go. So thanks, everyone. And thanks for the great introduction. I've been to many of these consortium talks, so I always enjoy them. And so I wanted to talk today about some of our work to develop ultraviolet photodissociation, particularly in the context of top-down analysis of proteins. And so for those who have never heard of ultraviolet photodissociation today, or UVPD, we're using ultraviolet photons to energize ions, in particular proteins, and presumably to generate lots of fragment ions. And so this is just a part of my group. We're really interested in characterizing many types of biological molecules in terms of determining sequences, modifications, conformations. And so today I'm really just going to focus on proteins, but we also have a component that is using UVPD to characterize lipids and nucleic acids. And so really our goal is to characterize these molecules in terms of determining fragmentation patterns that allow us to characterize the entire structure. And so we're going to be using photons from a laser to energize and blast these molecules apart. And so let me just show some of the methods to make this work. Now, we've implemented this method on a number of different ion trapping systems. And here's one example here where this is Lumos type system. It's one of the thermo orbitrap systems. So we have electrospray here. And at the back end, there's a couple of linear ion traps. And they're nicely aligned so that we have attached a window back there so that we can introduce photons from the laser. And so when the ions are stored in either one of these traps, such as shown here, where I've expanded it, the ions are trapped, they're oscillating. We can then fire the laser and irradiate them with photons. Our favorite laser happens to be an excimer laser that, when filled with argon fluoride, generates photons that are 193 nanometers, which is more than 6 electron volts per photon. It's a fast process. It's a 5 nanosecond laser pulse, and the activation process is very fast. So what does this look like for a protein here? We've got green fluorescent protein as an example. It's about a 27,000 Dalton protein. And so this is what the UVPD spectrum would look like if we isolated the 31 plus charge state, applied one pulse. And it's a pretty congested spectrum. But if you expand, one of these regions, for example, from M over Z 626 to 644, you see lots of ion profiles emerge, and they can be assigned to these different fragment ions. And as you'll note here, we've color-coded them. You'll see that we were generating AX-type ions, the traditional BY that you might see from collisional activation, and CZ are the type of ions you would see usually from electron activation. Now, the reason that we get so many different types of fragment ions is illustrated on this little energy diagram, which we like to compare to collisional activation. And so this is a potential energy profile where our ion, let's say a protein, is going to be stable once it's electrosprayed. So we need to add energy to cause fragmentation. And so if we do collisional activation, it's usually a multi-step process where the ion is slowly heated through multiple collisions, and then we generate fragment ions through the lowest energy pathways, which happen to be BY type ions. For ultraviolet photodissociation, it's a much bigger energy deposition with each photon. So we elevate the ions to excited electronic states, and then they can access other pathways that aren't accessible through collisional activation. And that's how we generate these AX and CZ type ions. And if you're more on the physical chemistry spectrum, you might enjoy this energy diagram, which shows an ion in a ground electronic state. After absorbing a photon, goes to an excited electronic state. And for proteins, this use of 193 nanometer excites the pi to pi star, which is the amide chromophore. And proteins have many amide chromophores in their backbone. And so then it can dissociate directly from that excited state to generate the AX, maybe the CZ ions. But if it undergoes internal conversion back down to the ground state, then we can generate ions akin to the collisional activation types of ions. And here's our little energy, our little fragmentation diagram. If you've forgotten your AX, BY, and CZ ions, we're just cleaving different backbone positions between the amino acids. I'll just interject here. Some people say, could we use a different type of laser? For example, how about using an IR laser? Some people have CO2 lasers, which are infrared lasers that generate 10.6 micron photons. That could also be used for photodissociation, and some people have used that. The photons are much lower in energy. They're only about 1/10 of an eV, so we never get to excited electronic states. It's going to require dozens, if not hundreds, of infrared photons to activate a protein. And you're typically going to cleave the most labile bonds just like collisional activation. And so with UV photons, you're getting a different type of excitation process. And really, when we think about this top-down approach, the challenge that we might encounter is that with larger proteins, you might only generate a few fragment ions, like this one, which shows 12% coverage, which is the number of backbone positions that are cleaved. That might be sufficient for identifying the protein, but our aim is to really characterize a protein, which means we need more extensive fragmentation that would allow us to localize modifications, such as if this lysine here was methylated or dimethylated. We need to be able to characterize the mass shifts associated with that, and that means we need a lot of fragmentation on either side of that modified site. And so that's a bigger challenge, and there's several obstacles that we're going to encounter to overcome these issues to get this very high fragmentation. And as we know with analyzing proteins, they're much larger molecules, and so the fragmentation is going to be split across many more fragmentation pathways. And so a smaller protein like this one, we might generate over 1,000 possible fragment ions with UVPD. That's assuming just 6 ion types. I haven't even talked about internal ions or hydrogen shift ions, and those can complicate the fragmentation even more. But when you go to a larger protein, you could have thousands of potentially different fragmentation pathways, and that means that you're really diluting your ion current. And then we have charge capacity limitations, which many people have noted in the past. For example, with ion traps, for example, maybe we can store a certain number of ions, but really it's based on the number of charges that you can store. And so when you have these multi-charged proteins, they occupy more of the charge capacity. And so for a lower charge state protein, you might be able to store 1,000 molecules, but if it's a higher charge state, which we often generate with these larger proteins in denaturing solutions, you might only have 25,000 molecules that can be stored. So you're dealing with fewer molecules to begin with. And then, especially with UVPD, there's spectral congestion. These large proteins are going to create very complicated spectra with hundreds, if not thousands, of different fragment ions. And so many of the ions are going to overlap with each other, and that will create an impediment to assigning all those fragment ions. And, you know, that makes me wish to reiterate here that when we're assigning these fragment ions, we really need these good isotope distributions. And so here I show a nice experimental isotope distribution for a Z ion. Well, we're matching that to a theoretical isotope distribution in order to assign that ion. And so you can see this fits well between the experimental and theoretical, but you can imagine if you have overlapping or truncated patterns, it's going to limit your ability to assign the ions. And that's a huge problem when we're trying to do confident characterization. And so getting back to some data here, here's an example of a UVPD spectrum for one of our favorite proteins, carbonic anhydrase. It's like a 29,000 Dalton protein, so not huge, but we can generate this type of fragmentation pattern using a single laser pulse. And right away we can see, we can assign ions to generate 65% sequence coverage, which is admirable. And, that's a good starting point. But we also know there's regions of the spectrum that are very dense, and there's, untapped resources of ions in these sections. For example, here we have many overlapping ions. The isotope distributions are overlapping also, and so we're probably not identifying as many ions as we could. And so we'd like to overcome that challenge. Oh, here's just an expansion of that where you can see in more detail. You can probably imagine that you see isotope patterns in there, but there's lots of intervening ions. There might be picket fencing going on, so assignments can be tricky. And so these are some of the obstacles we need to overcome with UVPD to further improve its performance for even larger proteins. And so we're dealing with sensitivity, signal and noise issues, and the congestion of the spectra. And so one way that we can address this is a technique that that others have adopted, and we've also applied it with wonderful success for UVPD, and that's charge reduction methods. And this has been commercialized on a number of mass spectrometers, and we feel like it's a game changer for UVPD because it's really a nice, we think, fairly easy technique to implement. So it couples well with the greater challenges of UVPD. And so in this method, if you have overlapping ions or overlapping isotope profiles for ions in different charge states and different assignments, if you can reduce their charge states through some type of reaction, they would spread out in the M/Z space. And this could mean a congested section of a fragmentation pattern could be dispersed and allow you then to assign those fragment ions. And we can do this with an ion-ion reaction called proton transfer charge reduction. And it's a super method to help sort out some of these ions. Now, this method, I will give credit to Scott McClucky's group at Purdue for pioneering this method. And really, it's now been adapted for many platforms with great success. And just to show it in greater detail, what's happening here is you take a proton scavenging reagent ion. It's going to be an anion because this is an ion-ion type of reaction. And this can then interact with fragment ions. For example, this A89 ion in the 10+ charge state. It can steal a proton, maybe from a protonated lysine side chain. It happens quite quickly in a few milliseconds. And our product would be a neutral reagent, which no longer we don't worry about, and a fragment ion that's lost one proton. And so it's still an A89 ion, but now it's been shifted by one charge. And so its mass to charge is going to be different. So it moves to a higher M/Z range. And hopefully it's shifted away from other overlapping ions. And so here's an example of a snippet of a mass spectrum on top for UVPD. We have lots of overlapping ions. Some potentially could be assigned. But if we do a proton transfer charge reduction reaction, this helps shift some of the ions to different M/Z values, and it really cleans up the quality of the spectrum. And so to show how this would be done on an instrument, for example, like this Orbitrap instrument, we start out the same way we would normally do with a top-down experiment. We're using electrospray to generate the ions. We transfer those proteins to the ion trap, the linear ion traps. We do the UVPD in the high pressure ion trap, and this creates a UVPD mass spectrum. Now we could stop right there and send the fragments to the Orbitrap for analysis, or we can engage the ion-ion reaction. And this is where we'll introduce our proton transfer reagent, send it to the back, allow it to overlap and interact with the fragment ions that we've already generated. And then we'll see the mass shifting as the original fragment ions start moving to higher M/Z space. And so here's an expansion again to show you our original UVPD mass spectrum. And then after 10 milliseconds of proton transfer charge reduction, we see more ions emerging at higher M/Z. And so some of these ions simply got reduced, and now they're showing up in higher M/Z space. And here's just one more example. Here's a congested portion of a UVPD spectrum. This is very typical for UVPD, lots of overlapping ions. Some of them have bad isotope profiles, and so we can't identify them. But after the proton transfer charge reduction, here's an example where now many more isotopically resolved profiles emerge. And then we can assign some of these ions that were questionable or discarded, and now they show up cleanly and can be identified. Now, what we've seen with this method is that we get much greater dividends if we start doing a method called fractionation. And this is where we look at portions of the spectrum and apply proton transfer charge reduction rather than apply it to the entire UVPD spectrum. And one of the major reasons we do this is because with UVPD, oftentimes we have a large portion of non-dissociated precursor ion. And we do that for a number of reasons, in part because we don't want to over-dissociate and cause too much secondary dissociation. But in any case, we have this very large, abundant, undissociated precursor, and that's occupying a large portion of the charge capacity of our linear ion traps. And then when we transfer those ions to the C trap, it has an even lower capacity. And so we're saturating our C trap with this uninformative, non-dissociated precursor. And so if we can do a section or fractionation, we can eliminate that precursor by isolating regions where there are going to be useful fragment ions and not transferring the precursor for analysis. And so here, this is a fractionation step where we looked at this region below or lower than the precursor. And we're already generating cleaner profiles because we've eliminated that very dominant precursor ion that causes charge capacity problems and dephasing of other ions. And then we do the proton transfer charge reduction, and we see now these ions start dispersing over the broader M/Z landscape. And you can see how this changes our coverage from 22% sequence coverage for the entire spectrum to 35% just based on fractionation, and up to 66% when we start incorporating the proton transfer charge reduction. So it's a stepwise process that is quite effective. And to show the workflow in more detail, we do this in sections. So here's our UVPD spectrum, very congested. We look at the lower M/Z range and do a proton transfer charge reduction step. Then we look at the higher range and do a proton transfer charge reduction step. Then we combine them to generate the most informative fragmentation pattern. Again, just to show this with real data, here's the lower region that was isolated and subjected to proton transfer charge reduction. Here's the higher region. And in each case, we're harvesting far more sequence coverage than if we just looked at UVPD alone. Okay, so now I wanted to show an example where we step through this one more time, where we look at a larger protein like enolase. Proteins, $30,000 and lower than that are, relatively straightforward to analyze by top-down analysis. But as we go to larger proteins, when we get the 50 kilodalton region and higher, it gets trickier. So here's enolase with about 46,000 Dalton protein. We isolated the 55 plus charge state. And with UVPD, one pulse, we have 17% coverage. Not super great, but it's a good start. And usually it's better than we can achieve with collisional activation methods. So, we think this is a winning spectrum, but it can get better. And so if we just do proton transfer charge reduction on the entire spectrum, this is the outcome. We're definitely seeing more ions emerge and they're extending to higher M/Z range, but we also see this very characteristic profile, which many of you will recognize is simply the precursor ion that's now been charge reduced to different charge states. So it's still occupying a large portion of the capacity of our trap. And we've improved our coverage to 43%, but we think we can do better. And so that's where we do our fractionation. We look at the lower range and do PTCR. We look at the higher range and do PTCR and combine all the ions, and we generate this map. It's much more filled in, and we see now we've reached almost 80% coverage. So this is definitely a step up. And I will emphasize that in our hands, the proton transfer charge reduction is really easy to perform. It's very successful, very robust, not nearly as challenging as the UVPD process itself. So it's a nice add-on bonus that's readily easy to do. And so, I've showed nice coverages and everything, but, attaining high coverage is one thing. We'd really like to be able to do this to analyze where modifications are and localize them. Because if we're just interested in coverage, in some cases, maybe you could do a bottom-up experiment and get very nice coverage too and identify more proteins. So if we're going to go to this high coverage, we really want to be able to put it to good use for identifying modifications or other things that happen to proteins, mutations. And so this little example I'll show is an example undertaken by grad student Jada Walker, where she was very interested in looking at ligands that bind to this protein New Delhi metallobeta-lactamase, 25,000 Dalton proteins, so it's not huge, but it's a super important protein because it turns out that some of our bacteria, like gram-negative bacteria, are producing this protein, New Delhi metallobeta-lactamase, or NDM, and it confers antibiotic resistance to the bacteria because this protein is notorious for hydrolyzing carbapenems, which are a super potent antibiotic, and it's often known as antibiotic of last resort for people stricken with these antibiotic resistant infections. And so it's important that we start developing better antibiotics or inhibitors for these, proteins that are really wreaking havoc on some of these bacteria. And so one of our collaborators, Chris Whitman, has been developing some of these inhibitors. And this is actually a covalent inhibitor. So it binds to the NDM protein. And they've made it selective for some of the nucleophilic sites, but this protein has 7 lysines and 1 cysteine, and they were obtaining conflicting feedback about where this inhibitor was binding to the protein. So they wanted to look at different approaches for really localizing that binding site. So this seemed like a great opportunity for top-down analysis, look at the intact protein and see where it's bound. And so here's the electrospray mass spectrum for the protein alone. It gives a very nice charge state distribution. And if you wanted to, you could use top-down analysis on the protein. But when it's the protein is incubated with the inhibitor, it undergoes a very efficient reaction and generates the inhibitor complex, which I've outlined in the red circles, showing it also gives a very nice charge state distribution. And we can select any of those complexes to do the top-down analysis. And so in this case, Jada isolated the 27 plus charge state. You can see, this is still a mixture of the apoprotein and the inhibitor bound protein. If you look at this little segment in the range of master charge 920 or so, you can see both forms are still there. She's just isolating this one with the inhibitor attached, and that's what will be used for the top-down analysis. And so here's the UVPD spectrum shown here. Again, we see the characteristic lots of fragment ions. It's congested. You can deconvolve it and start assigning many of these fragment ions. And for this spectrum shown here, she achieved a sequence coverage of about 58%. And that's pretty good for this protein. But of course, we're trying to pinpoint the binding site of the inhibitor. So we need to really bracket all those lysines and cysteines so we know which one ends up with the modification. And so in this case, here, she's fractionated the original UVPD mass spectrum into 4 sections and applied the proton transfer charge reduction to each of these sections. Section 1, 2, 3, and 4, carefully avoiding the precursor ion, which is simply going to undergo charge reduction and saturate the C-trap with uninformative ions. And so she's generating fragment ions from all of these 4 windows, and then she'll recombine them to generate the final mass spectrum. And so this is just a comparison. The UVPD spectrum shown, the pattern shown on the left, the UVPD plus the proton transfer charge reduction on the right. Sequence coverage has gone up considerably, still not perfect. We probably could use more sections of proton transfer charge reduction. But in fact, there's a few ions here that are really useful. And I'm not sure if you can look at this quickly and see where all the new fragmentation occurs. But we were honing in on this region around the cysteine and lysines, where we knew we were expecting this inhibitor to bind. And if we look at this little snippet of the UVPD spectrum, where there were some overlapping ions here, we were able to assign a couple of Z ions with good confidence, but there's some other ones, lower abundance, that were obscured, and we didn't have confidence assignments. So the 5 millisecond of proton transfer charge reduction really cleans this up. And for example, this unassigned ion here, which had kind of a pseudo profile, not perfect, pulls out fairly cleanly when it moves up to the 6+ charge state. And so now this ion can be assigned. And so you can do this step by step and start assigning more of those fragment ions, some of which are going to be quite informative. And in fact, this X60 was a real winner because it turned out to be a site right here. The X ions come from the C terminal end. So if you count up, this is where the X60 shows up. And this was a key ion because it bracketed this lysine site distinguished from the cysteine site that was close by. And so it turns out that for this case, the mass shift associated with this inhibitor was then localized to the cysteine site, not the nearby lysine. And this was important for understanding the process of this inhibitor attachment to this protein. So it was a real case where the top-down method provided important insight. Now, people often ask, with this UVPD, can we use it in an LC mode? LC mode and a top faster throughput time frame. And so I just wanted to show a couple of examples because you can implement this on a LC time scale. This is a study undertaken by Jada where she was looking at histones. Now, histones are not the largest proteins. So if you're just trying to identify or sequence them, they have some of the smaller sequences, so you can get very nice sequence coverage. But when you're looking at these are highly modified proteins that help, wrap around the DNA. So this coding of methylation and acetylation really is important for modulating the transcription of the nucleic acids. So it's important to understand the sites of methylation and acetylation. So this is a nice challenge for any kind of top-down method. And we see, chromatography doesn't look great because we have so many different proteoforms that are eluting. But, you can see that some of these profiles show extensive modification of each of these 4 classes of histones. And the UVPD allows, in some cases, very excellent characterization so that we can localize modifications that, in some cases we could not with collisional activation. Sometimes collisional activation outperforms UVPD. In many other cases, UVPD for this type of application where you're looking for modification analysis, it can work well. Now, more recently, we've looked at other types of larger proteins like antibody drug conjugates. These can also be analyzed in an LC mode. This was where the antibody drug conjugate was reduced and deglycosylated. So that separated the heavy chain, which is 50 kilodaltons, from the light chain, which is 25 kilodaltons, and then allows nice analysis of the molecular mass profiles, and then combining that with UVPD and proton transfer charge reduction, we can start getting quite nicely filled in fragmentation patterns for some of these modified antibodies. Now, the last couple minutes, I just want to give a little snippet to say we can extend this technique to even larger complexes. So I won't go into detail about this, but this was a recent study where we were looking at this multimeric protein complex, the 20S proteasome. This was a study we collaborated with Andreas Matuszek, who's here in our molecular biology department, and he specializes in understanding mechanisms of the proteasome. So his group nicely expressed and isolated some of the 20S proteasome for us. We now implemented UVPD on a different platform. This is on one of the ultra high mass range Orbitrap systems because we're looking at significantly larger protein complexes. And so this is over 700,000 Daltons. And now we're doing UVPD in an HCD cell, which is at the back end of this Orbitrap type instrument. And this proteasome consists of 4 stacks, or a stack of 4 rings known as the alpha and beta rings, which consists of these different proteins within those rings. And so it's a total of 28 different proteins in this protein complex. And so when this was Jada's work again, where she was analyzing there's the MS spectrum for this 20S complex. It sprays very well, and many groups have looked at this 20S proteasome, but it gives a very nice electrospray profile. She can isolate the charge states of several charge states and then do UVPD on that entire complex. And this is what's generated. Most of the ions show up down here. And if you expand this region of the mass spectrum, this is what it looks like. You can see many different proteins have been ejected from this complex. And she can start assigning them based on deconvoluting and see which proteins are released upon UVPD of this big complex. And so here's the deconvoluted spectrum showing that a number of the alpha proteins, which are the outer rings, are the ones that are preferentially released. And I won't dwell on it here, but there's a lot of other little ions here, lower abundance ions in this region. Those are actually fragment ions directly from some of these proteins. So you can actually start doing a little sequence characterization of some of these proteins that are released. Sequence coverage is low, but some of these ions are real fragment ions. And so, you know, she started looking at some of the different proteins that are released from these intact assemblies. And it's interesting to us that, first of all, the alpha proteins are preferentially released for some of these high charge state complexes. But when Jada used a reducing agent to lower the charge states, you know, by using something like triethyl ammonium acetate, it generates lower charge states, and then the disassembly of the protein complexes happens in a different manner. And when you see the charge reduction occurring, you generate some of these beta proteins are released, which are from the inner rings instead of the outer stacks of rings. So this is interesting, and it definitely deserves more attention. It's just another interesting example of how we can start using some of these techniques like ultraviolet photodissociation to look at some of these other types of protein problems. And so I think I want to quit there. I just want to say a few conclusions about UVPD. You've heard some of this before, but we are generating these very rich fragmentation patterns. It works well for many different charge states of proteins or protein assemblies. Because of the extensive coverage, especially when we pair it with proton transfer charge reduction, it really allows us to start pinpointing sites of modifications in more detail, which is a key goal of some of these top-down workflows. And I want to mention that a lot of this work is collaborative, and that's so important for many of these projects because we are not a biological group, so we really rely on collaborators who can provide us with great proteins and protein problems or other types of biological molecules, for example, lipids or many other types. And we collaborate across the spectrum, you know, not only locally, but with other universities and industrial collaborators, all who've helped expand our scope of research problems. And finally, wanted to thank all the great graduate students in the group and undergraduates who've contributed to the work and have been innovators and really fearless in the lab in terms of implementing some of these methods, and our funding agencies for providing the work, and hopefully this will continue, and other external collaborators, particularly ones I might have mentioned today, and an extra little shout out to Neil Kelleher, who really got us going with the top-down method in adapting ProCite for the many unusual types of ions created by UVPD. That was a huge step up. So thanks to the Kelleher group and all his amazing leadership. And also thanks to the thermoscientific team who's provided advice and guidance and technical support in some of these modifications that we've done. So I'll quit there and leave time for questions.
Oh, thank you so much, Jenny. What an amazing talk. I see here the Broad Squad and then Broad Squad. I probably said it wrong. And that is when you have a last name like Agar, you get really jealous because nothing good rhymes with it. So if you can help with that. So everybody, you know, you can put your questions in the chat if you have any. While you're doing that, I suppose Paul may be able to unmute people. I don't know how that works, Paul. But while you're doing that, I was just so impressed with the, well, all of it, fractionation, but the covalent drug, that's really incredible. That's a big deal and could even end up in an IND application, you know, because it just shows the MOA. It's just very impressive. Isn't a question, just a kudos. Let's see, no questions. Wow, that's
It's
unusual.
just
Go ahead, Neil.
yeah, hi everybody. It's just so lucid. It's a very clear talk. I have a thought that I'd love to get your take on. You know, the, it's a high level thought around sequencing. That what you're showing, the development of UVPD over the years shows that you could do full sequencing or very close to it. And as you know, there's the rise of nanopores and other single molecule proteoform sequencing. And so I wonder how you view that, say in the next few years, if nanopores start to emerge as something that's, you know, beyond their little squiggles they generate now, which is where they're at. Yeah, have you thought much about that? Because I think your tech is one of the best bulwarks in that if it is to be a competition.
Yeah, I say bring it on, but no, I think the nanopore technology
Love
is
it.
super fascinating. So it'll be interesting to see how well it does with all types of modifications and so forth. Is that really going to be in their scope? But
Mm,
and that's why, you know, in particular with UVPD, we're trying to try to go beyond just sequencing. Especially, for example, we moved into the nucleic acid regime, and I don't think anything can beat some of the methods out there for just standard sequencing of nucleic acids. But when you get to the modifications and other things, that's really where it gets tricky. And that's where I personally view that UVPD has its biggest role. It's things that are modified, and you need to identify and pinpoint and localize those modifications where other methods may fail.
We do have a couple of questions coming in. One is from Nina, and I hope I say your last name correctly, Apen. Great research. For data analysis, is there a scoring based on isotopic distribution of fragment ions for confident assignment?
Yes, there is. And lately we've been using ProSite native and you know the various ProSite methods where they do have isotope scoring algorithms, um, and they've indicated some recommendations. We tend to use maybe higher thresholds to instill extra confidence in some of our assignments. So we might use a isotope score threshold of 0.65 or 0.7. That'll make more sense if you look at some of those algorithms. We use the error tolerances when we assign fragment ions. And another thing that we do, which is I think particularly important with UVPD, is we always do replication, and we only retain ions if they're identified in at least 2, if not 3 separate spectra or runs. And so you can get a lot of spurious ions, which may be real, but we don't necessarily want to pin things on spurious ions. So they have to be replicated. And you've seen with UVPD spectra how dense they are. So there are many of these very questionable ions. So having these extra scoring metrics and scoring thresholds, I think, improves our confidence even at the expense of throwing some things out.
Great. We have another question from Andrew Swansinger. Compliments your research and then asks if what the limitations are in applying deconvolution algorithms in UVPD.
Yeah, well, we've looked at a few different algorithms. The one that we used to be using extract a lot. Now we're using ProSite native for just about everything. And so there's definitely room for improvement, at least, you know, in my world of UVPD, to improve some of this deconvolution. Um, we're not experts in that field, so we haven't been able to develop any of our own algorithms in this way, but we think it's an important undertaking, whoever has expertise to, you know, further continue developing and applying more scoring metrics.
Yeah, wonderful. Sal Barakara thanks you for your presentation and then wants to know how feasible is it to obtain informative fragmentation spectra that reflect protein and RNA DNA backbones while still retaining some information about the protein nucleic acid interface.
Yes, I think that's a great that's a great opportunity. Uh, and many people have jumped into the arena of native mass spectrometry or structural biology applications where they use solution conditions that will retain or preserve some of these native-like structures, transfer them into the gas phase, and then begin to dissect them. And so I think with something like UVPD, because of the activation process, we can generate ions from both the protein and a nucleic acid. So presumably you could generate both at the same time, creating even more complicated spectra. But we could also look for fragment ions where a piece of the protein is still bound to a piece of the nucleic acid or whatnot. And we've done that for a couple proteins where they're bound to small pieces of DNA. Big complexes are going to be much more challenging, and we're going to have to really worry about false assignments because you have so many combinations of the fragment ions from the protein and the nucleic acid. So really figuring out which ones are real is going to be tricky, but I think it's definitely a future ongoing goal.
Well, that's great. And you answered the second part of the question, which are what would be the challenges with doing that. Um, so Ayanu Awatuya, I'm so sorry if I say your name wrong. I see you there. Hey. Um, I saw a bar chart in the presentation. Is that a comparison between the signals in UVPD and UVPD PTCR? I'm new to this research area. Anyways, I want to know where the signals in UVPD PTCR that had it exact corresponding signals in the lower state UVPD.
Okay, I'll take a stab at that. I'm not
Yeah.
sure I'll answer the right question, but I'll
Yeah.
answer a question. And
Perfect.
sometimes when after the proton transfer charge reduction, the ions then are shifted to higher M over Z. And because then they're dispersed, we can assign their charge state and their M over Z with confidence. And then once we know they're there, we can look back in the congested non-charge reduced spectrum and figure out what they probably were. So that one example I think I showed, I'll just race back there real quickly, I think, if I can get there. This one here, we knew after the charge reduction, this was a 6+ charge state. If we look back there now, we think that it's there, it's the 10+ charge state. So this ion, this X60, which was a 10+, ended up here. It could not be deconvoluted and assigned, but after charge reduction, it ends up in a 6+ charge state way up here. And so we can kind of, work backwards and figure out where some of these ions were coming from. But it's a challenging process that, we'd rather just have the final end result where we can deconvolute the ions. And maybe we don't worry too much about what their origins were, because some of these are never going to be assigned. And, this is we like the outcome. We don't necessarily have to go back to the original to figure out where it came from.
Thank you so much, Jenny. And so we have another from Nina. In two-stage PTCR, where in the instrument is the PTCR happening following fractionation?
PTCR is always happening in the high pressure linear ion trap. That's something that Thermo has implemented superbly, and it works very well in that high pressure trap. So the ions can be manipulated between the sections of that little trap to get the reagent ions and then the protein ions, and then have them overlap in the middle section. And so that's where all the action happens.
Okay. Neil Kelleher asks, and I'm paraphrasing, He says, I don't trust Ryan Julian's so-called selective no, I'm kidding. One, Neil says, wondering,
That's
wondering
close.
to get
That's a
your thoughts on Ryan Julian's very selective photodissociation method, which I've seen Neil's very impressed by, and so am I.
I'm super impressed with it too. It's like the opposite end of the spectrum. Instead of getting more, he wants less, but less very selectively. It's a cool idea. I love it. He shows how well you can identify things. Now, I don't know that he'll ever get to characterizing things with that, but for identifying proteins, I love the idea. I love the selectivity, and it seems like he's hit a great, you know, a great strategy using some of his radical, specialized chemistry. So I'm a big fan.
Wonderful. And then our looks like what will be our last question is from Paul. He says, have you considered or tried shorter wavelengths to see if you can get even more coverage? And since he doesn't specify the wavelength, I think he's talking about cosmic rays.
Yeah, well, there's um, a couple groups are using X-rays. Uh, so that's been utilized. Um, and before that, there was people using 157 nanometer photons from using fluorine gas. That was Jim Riley's group at Indiana. He passed away a few years ago, but he was the one who pioneered using 157 nanometer photons. That worked super well. So you can use shorter wavelengths. We like the 193 because the 193 nanometer because it doesn't require any special vacuum conditions like some of those others do that have a tendency to ionize components in air. So then you have to have your light source also in a vacuum. So with 193 nanometer lasers, those can sit out in the lab and you can have the beam just passing through air. So that makes it a little easier to implement.
Yeah, I guess my thought also is, what's your thought? Do you think that more energetic photons will give you more useful fragments, or just blow it up into a bunch of small pieces?
I think from what we've seen from some of those others, you're getting the same types, A, B, C, X, Y, Z, some with hydrogen shifts and so forth. So maybe it would give a few more side chain fragments if you wanted to, you know, look at isomeric side chains, and that's a possibility. And Thermo, of course, has the 213 nanometer laser on several of their commercial systems, and that's a very robust laser, lower cost laser. And 213 nanometer is a longer wavelength. It also works quite well for proteins. Maybe takes a little bit longer time because that laser is a little less powerful. But I think there's lots of opportunities with different wavelengths. And Ryan Julian goes up to, for his super selective dissociation, I think he uses 266 nanometers. So that's another great option.
Right, thanks.
All right. And then we have one more, and I think we better call it. It is just how much extra time does it take to do the PTCR in terms of, yeah.
Yeah, PTCR for high charge states is pretty fast. It can be 5 millisecond, 10 millisecond. So it's much faster than the time of an Orbitrap scan. For those Orbitrap people out there, you know that the Orbitrap scans themselves, the high res, high accuracy scans can take a long time. So adding a little 5 millisecond PTCR reaction on is not a big deal. So that's not the rate limiting step.
Okay, wonderful. Well, it's 12 o'clock, so we should call it in the interest of everyone's time. Everyone go down to your React keys and give a big bunch of hand gestures for a wonderful talk. There we go. Oh, there they are. They're everywhere. This is great. And look, that's amazing. Look, I've never seen anything like it. I really haven't. Dang, I wish I'd recorded the screen. Yeah, you had at least, well, here they're still coming, Jenny. This is amazing. All right. Well, thank you so much. And thanks everyone for attending.
Okay, thanks everyone.